CMO DPAC allows you to pattern cells with very high precision and culture them in 2D or 3D. This opens up opportunities to study cell-cell interactions and the morphogenesis of engineered tissues. The main advantage of this technique is that it's easy for other labs to adopt. It doesn't require a clean room, specialized equipment, or custom synthesized reagents.
Begin by dropping small drops of the positive photoresist onto the aldehyde slide using a disposable pipette. Spin the slide at 3000 RPM for 30 seconds using the spin coater. Then place it on a 100°C hot plate for 1.5 minutes to cross-link the photoresist. Remove the slide from the hot plate and place a photomask with the desired features on top of the slide. Weigh it down with a piece of glass and cover the entire setup in an opaque box. Expose the setup with a UV lamp for 2 minutes. Develop the slide by immersing it in the developer solution for 3 to 5 minutes. Then rinse away excess developer solution with water. Dry it under a stream of air or nitrogen. Observe the slide under a microscope to confirm the success of photolithography and store it in the dark. Add a droplet of 20 micromolar amine-modified oligo solution onto each photopatterned region of the slide, and spread the droplet gently across the entire region using a pipette tip, taking care not to scratch the slide. Bake the slide in a 65°C oven until the DNA solution has fully dried.